Okay, well, I'd like to thank the committee very much for the invitation to come here, and thank them even more for the task they've taken on. So just to start us off with which things I think we already all know. So gene drive then, is this a pointer? Yes. So gene drive then refers to the preferential inheritance of a particular gene from one generation to the next. So in the simplest case, if you've got like a plus and a minus gamete coming together to form a heterozygote, then when that heterozygote makes gametes itself, they would not do it in a 50-50 Mendelian ratio, but instead in a 70-30 or 95-5 ratio. And that means there's been an increase in the frequency of one allele, the plus allele in this case, from one generation to the next, just due to the pattern of gene transmission. All else being equal then, in the population as a whole, that gene would increase in frequency, and it could do so relatively rapidly, going from, let's say, 1% to 99% in 20 generations or so, given a 70-30 split. And this drive process then can even lead to the spread of genes that are harmful to the organism carrying them. And it's that property that makes them so attractive then as potential tools for pest control. You can think of then of gene drive as being a sort of fifth force in population genetics, in addition to the usual textbook cases of mutation, migration, drift, and selection. So there's many natural examples of driving genetic elements. So this is a natural phenomenon that has evolved many, many times in nature. So these are some of the best known examples of them. Transposable elements are found in virtually every organism that's been studied. Gamete killers, such as the T haplotype in mice, has been very well studied. Segregation distorter in Drosophila. Driving sex chromosomes have been described in both Drosophila, fruit flies, and in mosquitoes. There's also maternal effect killers that have been described from flower beetles, and homing endonuclease genes found in many microbes. There's many other examples that are less well known, perhaps, to people, but have nevertheless been described. So B chromosomes, neocentromeres, group 2 introns, maternal sex ratio-like elements, harbor genetic hemiclones, androgenetic elements, can mutate factors, and X-linked feminizers. So there's a whole zoo, a whole biodiversity, if you want, of these genetic elements that are able to spread through populations because they show this drive. Many of these ones have been less well studied. For many of them, the molecular mechanisms are less well known and could, in principle, be sources of novel approaches in the future as we learn the underlying biology. Now, as I think has been mentioned in previous presentations here or on webinars, the strategies or objectives for using synthetic gene drive systems for controlling pest populations can kind of be divided into two types. First of all, there's population suppression, and that could be used against vectors of diseases or agricultural pests or invasive species. And there you're targeting either the survival or the reproduction of the target organism. And when we're talking about reproduction, that could be the fertility, for example, or it could be the sex ratio. The alternative large class of potential interventions are population modification, which is usually talked about in the context of disease vectors and less so in the context of agricultural pests or invasives. There you can divide so this is where you're leaving the numbers of the pest species more or less intact, but changing them such that they're less harmful, less able to transmit the disease, for example. So again, we can split this into two different types, broadly speaking. So you can knock out a gene that's found naturally in the vector species and the target species, knock out a gene that renders it less harmful. Or you can try and introduce a new gene that renders the species less harmful. And there's different sorts of phenotypes that people have talked about and discussed and done some research into in this population modification umbrella. So you can, for vectors, for example, it's often the older age classes that are most harmful, most dangerous, most at risk of transmitting disease. And so if you can change the age profile of the population, then by targeting the older individuals, then that can reduce disease transmission. Or there's this idea that's been discussed that if you can selectively kill just those vectors that are infectious, then that would help reduce disease transmission. Or people have speculated about changing the host feeding preference of the individual, or, and this is the most common thing that is discussed, is to change the vector competence so that the species is no longer able to support the development of the pathogen. So there's many different types of things that have been discussed. And even for vectors where there's been the most discussion, I think it's premature to say which one of these is best. And I think all of them have the potential to contribute. And combinations of them are also possible. In terms of synthetic man-made gene drive systems for control, there are 4 different types that have been received the most attention. So there's the homing type of reaction. It's also recently been renamed as the mutational chain reaction. Then there's sex chromosome drive. There's the killer rescue systems like Medea, and then there's underdominant systems. And each of these have their different uses and features. So for population suppression, for example, both the homing agents, homing elements, and the sex chromosome drive can, in principle, be used and are being, people are trying to develop them for population suppression. I've not heard anybody, and it's not clear to me that you could use the killer rescue systems or the underdominants as drive systems in order to suppress a population. But there might be people working on that, but I think it's received somewhat less attention. For population modification, again, we can divide it into the knockouts and the knock-ins. For the knockout, where you're trying to eliminate a gene in the target species, then the homing is kind of the simplest one. You would do a population-wide knockout using a homing element. In principle, you can use these other ones, but it's quite a bit more complicated. It's not less straightforward. Or to knock in, so all of the different types could be used to knock in a gene that makes the species less harmful. These different approaches of suppression versus modification raise different sorts of issues, and so have different sorts of features. So obviously in one case you're reducing the numbers of the organism or leaving the numbers intact and changing how harmful they are. So in terms of the equilibrium abundance of the construct then, once you have rolled out your intervention, under population suppression, the construct is either in low abundance or not present at all, whereas with population modification, your construct is at high abundance. In terms of the propensity to self-extinguish or burn itself out, that's a feature or property, if you want, that will sometimes occur with the population suppression approaches, is unlikely to occur with the modification ones. And similarly for competitive release, where you're changing the numbers of one species, that may have in some circumstances effects on other species. For the population suppression, again, that's going to be less likely of an issue with the modification. In terms of the evolutionary stability of the construct, so thinking about once you release something, what's going to happen when mutations arise in that construct which affect its functioning. With the population suppression ones, if you get mutations that destroy the drive, destroy the homing, or destroy the sex range distortion, those mutations are expected to disappear and not accumulate. Whereas for the population modification, if you get mutations and you're trying to knock in a gene, an effector gene, then mutations that destroy that effector function may be selected for, leading to spread then of an ineffective or empty construct. And so that puts the constraint of having to have effectors in there that you're driving in with small fitness effects. Now, in terms of opportunities to spread counteracting constructs, things to counteract the original construct that you put in, you can, for population suppression, you're putting a load upon the population, and you can use natural selection to spread a counteracting construct. Whereas for the population suppression one, for the population modification, since effectors are going to have small fitness effects, then counteracting that small fitness effect will also have a small fitness effect. And so you cannot use natural selection, you'd have to rely on drive to spread a counteracting construct. And in terms of the thinking about in any of these things, if you're going to try to control pest or disease population, you have to think about resistance. And in these two cases, the target of selection for resistance is going to be different. On the one hand, it will be the vector species or the pest species. On the other hand, it will be the pathogen itself. So the different approaches then raise different issues. And I think, this is just a very high-level thing. In order to judge, it's going to be very important to judge any specific instance that it be treated on a case-by-case basis. So these counteracting, just a few words about these counteracting constructs that have also been called reversal constructs or recall constructs or remediation constructs. I'm not really sure what the best phrase for them is. Personally, I say that I started out quite enthusiastic about the idea. I'm now somewhat a little bit more ambivalent about them. And the critical point, I think, is that when you do risk assessment and risk management, that you not rely on them. So they're not any kind of substitute for a thorough risk analysis at each step. And it's really important not to have a cavalier attitude towards risk that say, oh, we can release something, and if anything goes wrong, well, we'll just release something else that counteracts it. So we certainly must avoid that. And indeed, I think the first choice is to be able to mitigate the risks to such an extent, to an acceptable level, without having to consider them. I'll just give a couple slides on each of these two, the homing and the sex chromosome drive that we've worked on the most, and so I know more about. The homing approach, the homing reaction, that's this one where you've got a gene for an enzyme that recognizes chromosomes not containing the enzyme, cuts it, and then gets copied across during the process of repair of this chromosome, converting a heterozygote to a homozygote. So the idea is, or the simplest way of using this, is to have a nuclease gene inside some target gene, disrupting that target gene, and then it recognizes a sequence in the empty target gene, cuts it, gets copied across during the repair process, and so you can get a heterozygote converted into a homozygote knockout of that target gene. So this is based on naturally occurring homing endonuclease genes that exist in fungi and algae and other microbes. And so you can get this population-wide knockout of a target gene. You can also use this approach to knock in a gene, for which there's a couple different alternative strategies, but they're a little more complex than just knocking out a gene. There's different enzyme architectures that can be used for doing this. There is the naturally occurring homing endonuclease architecture that you could change a fungal or algal homing endonuclease to recognize a mosquito sequence, for example. Or there's various sorts of synthetic ones. So there's zinc finger nucleases, tail nucleases, and the CRISPRs, which have gotten a lot of attention recently. And we seem to get a new architecture every 5 years or so. So 5 years from now, there could well be other ones to be adding on the list here. CRISPRs have been getting a great deal. There's a number of criteria to be thinking about when you're deciding about these alternative architectures. So ease of engineering is a really important one, obviously, to be able to target a specific sequence in a mosquito or whatever species. And that's why CRISPRs have gotten a great deal of attention recently, is because they are so easy to re-engineer. But that's not the only criteria that's going to be important in thinking about a product for actual release and rollout. So the other sorts of criteria we need to be thinking about, of course, the enzyme has to be active, has to have good specificity, so it targets what you want to target and not elsewhere in the genome. It's important what kind of DNA repair is stimulated by the different sorts of enzymes. They leave different kinds of jagged ends, different types of cut sites, and so that can stimulate different types of repair processes by the organism, and that's important in thinking about these things. Is the stability of the activity and specificity. So as these were to spread through a population, mutations will accumulate, and so one needs to consider how those mutations will affect the activity and the specificity of the enzymes. And also very important, as I mentioned, resistance, thinking about that's important, and so the robustness of these various architectures to resistant alleles is going to be critical. So again, I think it's premature to say which one of these different architectures is going to be best in the organisms of applied interest. And all have potential to contribute, and combinations are also going to be possible. The driving Y chromosome approach is based on cleavage without homing. So it's the idea of having put a gene for an enzyme on the Y chromosome that's specifically active when the male is making its sperm. And so then that would recognize a sequence that's only found on the X chromosome, ideally a repeated sequence. And then it shreds up the X chromosome when the male is making its sperm. And so the only functional sperm that are produced are those carrying the Y chromosome. And those will also carry the gene. And so that gene, that Y chromosome, gets to 95% of the progeny instead of 50%, the Mendelian 50%, and so is able to spread through a population. So again, this is based on a naturally occurring driving Y element found in Aedes mosquitoes, which we don't know the molecular mechanism behind it, but we do know that the X chromosome breaks at meiosis. And we've got proof of principle that have been reported in Gambiae, Anopheles gambiae, that give 95% males with normal fertility. So the idea here, just in very cartoon, is to have a population, starting a population 50-50 males and females. You introduce a relatively small number of modified males carrying this driving Y chromosome, and then over successive generations, the driving Y chromosome carrying males tend to become a larger and larger fraction of the male population, and males as a whole become a larger and larger fraction, and females become more and more rare within the population, until such a point that you've only got males, or there's so few females to support the population, and you get local elimination. That's the case if you've got like 100% drive. If you've got a sort of weaker drive, then that can lead to suppression of the population rather than elimination. Okay, it doesn't matter, sorry. That feature of being able to change, that the outcome will depend, suppression versus elimination depends upon how strong the drive is an example of something that is important to bear in mind, is that even the simplest system of a single gene causing a driving Y chromosome or a single gene causing a homing event, that has a number of features about it that you can change, which will change the population dynamic consequences. So enzyme expression or activity, changing that can make the difference between population suppression versus elimination. Changing the half-life of the enzyme, we have found, can change the results of can lead to a male being either sterile or fertile, and so that can lead to either immediate disappearance or gradual disappearance of a construct from a population. And then you can change the promoter, make it constitutively active or active only in the germline. You can have a promoter that's active before or after the target gene in the context of the homing endonucleases. And that will change the dynamics from either being self-limiting, that is to say, not spreading once you release it and gradually disappearing, versus self-sustaining, spreading from a small inoculative construct. And just like, in addition to changing the promoter, you can also change the target gene, the gene that you're targeting in the case of homing, whether the knockout is dominant or recessive. That will again change the dynamics and the location of the construct. So even in this very simplest system, one-component systems, there's multiple little bits that you can change, and multiple component systems will be even more complex. And a nice feature of this is that it allows a step-by-step development pathway. So you can then have a step-by-step succession of modified mosquito strains, starting, for example, with sterile males, then progressing to fertile but self-limiting strains, and then finally a self-sustaining with gene drive. So there's a progression of constructs of increasing effectiveness. And then on each one of them, you will want to do a succession of studies, doing them offshore, for example, in the lab, then in-country in the lab, and then in small-scale field releases. And so at each step, then, there will be technical, regulatory, risk, and engagement activities which can inform the next step. And finally, I'd like just to end with a couple notes on precedents. So this gene drive approach hasn't been done before. So we can't say, oh, there's a well-trodden path for this. But there are nevertheless some things out there which I think we can get useful lessons from. And so in terms of containment, Well, we want to keep gene-drive things in the lab. Well, this is not the first time we've had this issue, and infectious agents or exotic species we also want to keep in containment, and so we can learn from that. In terms of the releases, probably the best parallel is conventional biological control programs. So this is just an example. This is not the malaria belt in Africa, but it's very similar to it. It's where the cassava mealybug was in the 70s and 80s. Cassava is a staple crop from South America. At some point, this mealybug got introduced from South America, caused all sorts of devastation. And the product that was used to control that was this wasp that was brought over from South America, released in multiple places in sub-Saharan Africa, released with the intent that it would then spread and suppress the population that it was meant to target. So it was released in various places around sub-Saharan Africa, and it had a great impact in reducing the problem of the cassava mealybug, and with a very high benefit to cost ratio. So that's sort of an example that we keep in mind when thinking about the step of releasing a gene drive system. This is not just releasing a single gene or one or two genes, this is releasing a whole organism, and they found a way to do it, and it can be very useful. So I just want to conclude with a thank you for your attention, and a thanks to participants in the Target Malaria program, which is a not-for-profit research consortium that I'm part of. There's too many people to be involved, but that's a list of the various institutions. Thanks very much.